Hello everyone. Uh, Good afternoon everyone. Again, synthesis of fatty acid. Just I'm welcoming all right now for the second step of biosynthesis of the fatty acid. Biosynthesis, will just revise something. Biosynthesis of fatty acid is also known as a de novo synthesis of fatty acid. Then it is also known to be happening in the liver, adipose tissue, kidney, locating in the mammary glands and cytosomal fraction of the cell. It has been happening. This required a carbon source for the fatty acid is coming from the acetyl CoA. Then NADPH is a reducing equivalent. Along with that, ATP is used as energy. Now, there are three stages of it. In last lecture, the stage 1 we had completed, that is the production of acetyl CoA and NADPH. Now we are moving towards the conversion of acetyl CoA to malonyl CoA. The third step, reactions of fatty acids in this complex. This is nothing but a 10-step reaction is nothing but the present for the third one. So here we are moving to the first revise of production of acetyl CoA and NADPH. When we're revising the acetyl CoA has been a byproduct of a product forming in the mitochondrial matrix by the oxidation of pyruvate acid atom amino acid fatty acid to form acetyl CoA. Then there is another product which is forming in the mitochondrial matrix is from the pyruvate is oxaloacetate. To form a citrate, because acetyl CoA is not permeable to the mitochondrial membrane, and we require acetyl CoA for the biosynthesis of fatty acid. The bifurcation pathway is there, that is nothing but acetyl CoA along with the oxaloacetate to form a citrate. A citrate is a permeable to the mitochondrial membrane. In cytosol, what is happening? This citrate, in the presence of citrate lyase, it will convert into acetyl CoA, which we require for the fatty acid synthesis. And along with that, ATP is used as energy. Then oxaloacetate has been second product, which is forming from the citrate. And along with that, malate dehydrogenase and the NADH and H plus. It is converted to NAD and it is giving hydrogen to form a malate. Then further a malic enzyme to form a pyruvate. These are the ongoing cyclic process. Now NADPH is requirement for the synthesis of biosynthesis of the fatty acid. We required acetyl CoA and also we require NADPH and CO2, which has been we are getting from the step 1. Now we are moving towards the step 2. When we think about the step 2 of now biosynthesis of fatty acid, step 2 is known to be conversion of acetyl CoA to malonyl CoA. Now in the step 2 biosynthesis of fatty acid is conversion of acetyl CoA to form a malonyl CoA. Now we are drawing the structure of acetyl CoA, that you know, CH3C11O SCoA. This is acetyl CoA. Now this acetyl CoA, the second step which we were forming, conversion of acetyl CoA to malonyl CoA, this one known as a carboxylation reaction. This carboxylation reaction required carbon dioxide is fixed with the help of enzyme. Now, this acetyl CoA is the CH3COSCoA, acetyl CoA, in the presence of acetyl CoA carboxylase. As you know, carboxylation process is nothing but we require fixation of carbon dioxide. In that we require carboxylase as an enzyme, and this enzyme also required a biotin. biosynthesis of fatty acid step 2, that is conversion of acetyl CoA to malonyl CoA. In this acetyl CoA will give rise to form a malonyl CoA. Malonyl CoA. And this acetyl CoA convert into a particularly malonyl CoA. In this reaction, acetyl CoA carboxylase is an enzyme which has been utilized in the presence of biotin, and CO2 is get fixed up, will convert into the energy is used, energy source which we are utilizing is ATP. Now acetyl CoA CO2 is get fixed up in the acetyl CoA, so where it has been there like this here. So product which you are forming is COO dash CH2 CO SCOA. So in this reaction we are getting acetyl CoA is converted into malonyl CoA by carboxylation reaction. In the presence of acetyl CoA, carboxylase and biotin is required. In this reaction, ATP used as an energy source and fixation of a carbon dioxide takes place. So this is nothing but the second step of the biosynthesis of fatty acid. Now, acetyl CoA carboxylase is known to be a regulating enzyme. So, for the biosynthesis of fatty acid, we required acetyl CoA carboxylase. So this is nothing but the reaction we required. These are the happening in this reaction. So again, we are repeating this one. The biosynthesis of fatty acid, the first step 2, in this conversion of acetyl CoA to malonyl CoA. This is your second step. In this, it is forming acetyl CoA. It is a purely carboxylation reaction. So this acetyl CoA convert into malonyl CoA in the presence of acetyl CoA carboxylase and biotin. ATP used as an energy source, and this ATP convert into ADP plus Pi, and carboxylation reaction. So we require the carbon dioxide has been fixed, and this is nothing but forming a product, a malonyl CoA reaction. So this step 2 is known to be carboxylation reaction, and rate limiting here is nothing but the enzyme regulating enzyme acetyl CoA carboxylase. So this is nothing but the step 2 of biosynthesis of fatty acid. Now again I am revising because in first lecture I got this as a request from the students that I have to repeat the first reaction and then we will continue with the second reaction. So first reaction is nothing but a biosynthesis of fatty acid. In this stage 1 is known to be production of acetyl CoA and NADPH. Whereas second step is known as a conversion of acetyl CoA to malonyl CoA. And third reaction is reaction of fatty acid synthase complex. Now when we think about the first reaction, as a source we required acetyl CoA for the synthesis of the fatty acid. So in mitochondrial matrix we know oxidation of amino acid, fatty acid, pyruvate is forming a product known as acetyl CoA. We require this acetyl CoA inside the cytosol for the formation of a fatty acid. But unfortunately acetyl CoA is impermeable, not permeable to the mitochondrial membrane. And that's why this should be converted into such a form so that it will go outside and again it can form regain its structure. So here the acetyl CoA is formed, the oxaloacetate. Together it is nothing but forming a citrate in the presence of citrate synthase. This citrate will go outside in cytosol and again it is converted to oxaloacetate in the presence of citrate lyase, and that acetyl CoA is formed for the fatty acid. Now this oxaloacetate further converted to malate in the presence of malate dehydrogenase, and this malate converted to pyruvate in the presence of malic enzyme. And NADPH is formed from the NADP, and CO2 is formed which has been utilized. Now the second step which is very importantly seen, so biosynthesis of fatty acid. Step 2 is conversion of acetyl CoA to the malonyl CoA. Here the acetyl CoA, this reaction is purely known as a carboxylation reaction. In this carbon dioxide fixed with the help of enzymes. And this acetyl CoA, CH3COA, CoA, that is acetyl CoA, in the presence of acetyl CoA carboxylase and biotin, it is converted into malonyl CoA. And this malonyl CoA is nothing but we can say by the fixation of carbon dioxide inside the acetyl CoA. Fixation is required, the energy source, that is nothing but ATP converted into ADP and PI, that is inorganic phosphate. And this is nothing but forming a malonyl CoA. Thank you very much for this time. Tomorrow we will learn about in the third step or step third or third video, we will move on to the next topic, next step of this biosynthesis of fatty acid, that is a reaction of fatty acid synthase complex. This is a 10-step reaction that we will learn tomorrow itself. Thank you guys.